All right, so as Adrian has been performing his flying hamster crosses, he's noticed that some of the hamsters have horns. And unfortunately, they keep headbutting him when he's not looking. And, finally he just gets fed up with it, and he decides he's going to create an experimental strain without any horns. But as he's looking through the hamsters, he realizes that all the males in all of his strains, doesn't matter which phenotype he was studying, they all have horns, and none of the females have horns. So in his previous experiments with coat color and ear size, all of the genotypes and the phenotypes in those cases were evenly distributed among the sexes. So now he's wondering, what could be going on with this whole horn thing. Adrian's puzzled, so he walks down the hall again to, have a discussion with his friend Ben and try to figure out what could be going on here with this horn gene. Ben again tells Adrian, that you really have to be careful about exceptions to scientific rules, and that, as he pointed out earlier, that there are lots of exceptions to Mendel's laws. So Ben explains that sex is determined in many organisms by what are called sex chromosomes. So the genotype of the sex chromosomes determines the sex of that individual. Now, how the sex chromosomes determine the sex of an organism varies from species to species. So in humans, the presence or absence of the Y chromosome determines the sex of the individual. So for instance, an XY individual is male and an XX individual is female. Now, if we draw out a Punnett square, we can actually see how the presence or absence of the X chromosome is determining the sexes. If we have XX mother and an XY father, we can see that we're going to get 50% XX individuals, which will be female, and 50% XY individuals that will be male. So it's interesting to note that the father is the one that determines the sex of the children. The only sex chromosomes that the mother can contribute are X's. So whether or not the child gets an X or a Y from the father is what determines the sex of the child. And it would have probably helped out Anne Boleyn a little bit if they had known a lot more about genetics back in her time, so she could have avoided her husband Henry VIII's wrath that she wasn't producing a male heir to the English throne. Now, since males only have one X chromosome, the sex chromosomes present a unique situation compared to the rest of the chromosomes in the genome. Now, the chromosomes in the genome which are not sex chromosomes are called autosomes. And up until now, we've only been considering genes that are on the autosomes. In the case of the autosomes, there are always two copies of the gene, one on each of the two homologs. And since there are two copies, a dominant allele can mask the phenotype of a recessive allele. So this is a major reason why people can be carriers for disadvantageous genes. So consider a disease like Sickle-Cell Anemia. It's caused by the presence of two recessive alleles. Now, in this disease, red blood cells can become sickle-shaped and lodge in small blood vessels. Right? So you can imagine this produces a whole host of problems for someone that's suffering from this disease. So let's examine the genetics of this disease a little bit. So let's represent the disease allele with a lowercase SCA, since it's the recessive allele. And let's represent the quote-unquote normal gene, or the non-disease gene, with an uppercase SCA, because it's the dominant allele. So if we have an individual that's little SCA over little SCA, that individual is going to exhibit the disease. So in contrast, if I have big SCA over big SCA, that individual does not exhibit the disease phenotype, because the big SCA allele is the non-disease allele. Interestingly, though, the individual that is heterozygous for the two alleles, so big SCA over little SCA, also does not exhibit the disease phenotype because the dominant allele can mask the phenotype of the recessive allele. And we refer to this person as a carrier because this person does not exhibit the phenotype but carries the ability to pass on that phenotype to his or her progeny. So let's now try to apply this concept of recessive disadvantageous traits to the sex chromosomes of humans. Since there are two X chromosomes in a female, a disadvantageous recessive phenotype can be masked in a heterozygote. This means that the recessive traits on the X chromosome are less likely to be observed in a female. However, males only have one X chromosome. So that means that whatever's on the X chromosome is going to get expressed because it's the only allele that can be expressed. So that means that males have a higher chance of displaying a recessive X-linked trait because there's no way to mask the recessive phenotype. Now, a recessive gene that is located on the X chromosome produces a sex-linked trait because a recessive trait will preferentially be found in males compared to females. A classic example of a sex-linked trait is colorblindness. Now, the colorblindness gene is located on the X chromosome, so men are more likely to be colorblind than women. Now, to understand how this is working a little bit better, let's return to our Punnett squares. So if we represent the colorblindness allele as little cb and the normal allele as big CB, we can track the genotypes and the phenotypes in our Punnett square. So let's consider the children of a woman who is heterozygous for the colorblind gene and a man who isn't colorblind. Okay, so how many girls are going to be colorblind? If we look at our Punnett square, we can see that both of these girls have at least one big CB allele. So none of the daughters are going to be colorblind because each of them has at least one good colorblindness gene. How many of the boys will be colorblind, though? Well, half of the sons are going to be colorblind because half of them are going to receive the recessive CB allele from their mother. So notice that the children of this carrier mother had the same chance of getting the colorblindness allele. However, since girls have two X chromosomes, a second good copy of the colorblindness gene can mask that phenotype. However, since the sons only have one X chromosome, half of the sons end up exhibiting the colorblindness phenotype. All right, so Adrian rushes back to the lab now with a new perspective on chromosomal-based gene inheritance. Since he never sees females with antlers, he speculates that maybe this is similar but not quite the same as the colorblindness example that Ben was telling him about. And since only males have horns, he hypothesizes that maybe the gene is on the Y chromosome. So if the gene was only on the Y chromosome, that could explain why only males ever exhibit the trait. Now note that in the case of a sex-linked trait like colorblindness, it is linked to the males, but is not exclusive to the males. In this case, we have a trait that is only expressed in one of the sexes. This is a sex-limited trait. Note, though, that this is an overly simplistic example where the sex-limited gene is on the Y chromosome. But sex-limited genes are usually found on an autosome. They're just only expressed in one of the two sexes. So in summary, we've learned that sex chromosomes are the chromosomes which determine the sex of an organism. Autosomes are the chromosomes in the genome which are not sex chromosomes. A carrier is an individual heterozygous for a recessive phenotype. A sex-linked trait is a phenotype produced by a recessive gene that is located on the X chromosome. A sex-limited trait is a trait which is only expressed in one of the sexes.